Welcome to the Deep Dive Lab, where we dive deep into fascinating science stories filled with surprising discoveries, unexpected twists, and thought-provoking ideas. When we look at nature, it has these remarkably elegant, almost counterintuitive ways of solving really complex problems.
Oh, absolutely.
And take metal, for example. When we think of metal, we typically picture macroscopic engineering, right? Like suspension bridges, skyscrapers, heavy machinery.
Right, big stuff.
Exactly. But biological systems use metal ions on a microscopic scale to build these incredibly precise functional structures.
It's a fascinating biological quirk, honestly. Nature essentially uses metals as the ultimate structural scaffolding. We see it everywhere once we actually start looking for it.
Yeah.
Like iron ions, for example, sitting perfectly inside our blood's hemoglobin, they create the exact chemical geometry required to bind and transport oxygen.
Which is crazy to think about.
Right. Or consider magnesium ions. They actually help orchestrate the crystallization of our tooth enamel. Yeah, they align the minerals to create the hardest tissue in the human body.
So it's kind of like a microscopic biological metalsmithing.
Yeah, that's a great way to put it.
And that brings us to the mission of today's deep dive. We're looking at a 2026 paper published in the journal Science by researchers at the University of Michigan and their collaborators. And they looked at that biological metalsmithing and asked this really brilliant mystery question. What if scientists could use this exact same trick to construct a microscopic Trojan horse? One that could, safely sneak into a tumor and sound the immune system's alarm against cancer.
Because sounding that alarm is arguably one of the most frustrating bottlenecks in modern cancer immunotherapy. We possess the biochemical alarm bell, but we just haven't known how to ring it safely.
Right.
The stakes are incredibly high here. For a patient with advanced cancer, stimulating the immune system too little does absolutely nothing, but stimulating it too much can trigger lethal systemic side effects.
Let's ground this in the human reality for a second. Think of the body as having a built-in fire alarm system for cellular danger.
Okay.
Historically, when doctors have tried to pull that alarm using these really powerful drugs, it hasn't acted like a targeted fire extinguisher just putting out a localized tumor.
No, not at all.
It's behaved more like setting off a massive sprinkler system that completely floods the entire building, destroying everything inside, just to put out a small fire in a trash can.
Yeah, that's exactly what happens.
So we need to understand how these scientists finally fixed the alarm. But to do that, we first have to understand the biological hardware they were trying to trigger, right?
Right. And the hardware in this case is called the cGAS-STING pathway. It's a fundamental component of our innate immune system. Basically, it acts as an internal cellular security guard looking for stray DNA.
I want to pause on that because I think the mechanics are crucial here. When we say stray DNA, we are talking about genetic material floating around in the cell's cytoplasm, right? Like outside of the nucleus where it actually belongs.
Exactly. That is the trigger. DNA belongs safely locked inside the nucleus. So the first half of that pathway, cGAS, which stands for cyclic GMP AMP synthase, is the actual sensor molecule.
Okay.
If cGAS detects DNA floating freely in the cytoplasm, it knows a catastrophic event is occurring.
Like what?
Well, it means either a virus has invaded and is replicating, or the cell itself is breaking down and becoming cancerous.
Oh, I see. So cGAS spots the debris on the factory floor, realizes there's an emergency, and then what happens? It passes the message to STING.
Yeah, it creates a messenger molecule that physically binds to the STING protein, and STING stands for stimulator of interferon genes.
Okay, got it.
Once STING is activated, it triggers the release of interferons.
Yeah.
Think of interferons as a biochemical battle cry.
A battle cry.
Yeah, they flood out of the cell and recruit the rest of the immune system's heavy artillery to come in and clear out the danger.
And scientists have known for a while that if you can intentionally activate STING, you can absolutely obliterate tumors, right? The original belief in the field was incredibly optimistic. The logic was basically, hey, we have these drugs, these small molecule STING agonists or activators. If a patient has advanced cancer that has metastasized, let's just inject these STING activators into the bloodstream and let them hunt down tumors all over the body.
Yeah, the logic was sound on paper, but the biological reality was a severe bottleneck.
Because of the toxicity.
Exactly. When you inject free STING-activating drugs into the bloodstream, they get cleared out of the body very quickly by the kidneys and liver. So to get enough of the drug to actually reach and penetrate a tumor, you have to pump the patient full of massive, continuous doses.
Which brings us back to the flooded building analogy. Those massive doses cause Widespread immune toxicity.
Lethal toxicity in many cases.
Wow.
Yeah, you're triggering systemic inflammation. The interferons are essentially telling every single cell in the body that it is under severe attack,
Oh my gosh.
which leads to tissue damage and organ failure.
But if the drugs are that toxic when they're just freely circulating in the blood, the obvious pushback from, a clinical perspective is, why didn't doctors just inject these S-staining drugs directly into the tumors themselves? Just bypass the bloodstream entirely and keep the fire extinguisher localized.
Well, they certainly tried that, but intratumoral injections only produced modest results.
Really?
Yeah, the local tumor might shrink, but it does absolutely nothing for cancers that have metastasized to the lungs or the brain or the bone.
Oh, right, because they're everywhere.
Right. You can't physically find and inject a needle into every microscopic tumor site in a patient's body.
That makes sense. But wait, what about traditional lipid nanoparticles? We hear a lot about those little fat bubbles used in mRNA vaccines to protect the fragile cargo. Didn't they try packaging the drug in those?
They did. But for delivering S-STING drugs to tumors, they completely failed.
Why?
The body's filtration system is just too efficient. When traditional squishy lipid nanoparticles enter the bloodstream, the liver treats them like cellular garbage.
Oh.
Specialized cells in the liver, called Kupffer cells, basically sweep them up. So the nanoparticles just accumulated in the liver and failed entirely to trigger a strong immune response inside the actual tumors.
So the field was just stuck.
Completely stuck. The drugs were either too toxic in their free form, or they got totally lost in the liver when they were packaged.
Which brings us to the University of Michigan researchers and their Trojan horse.
Right.
They hypothesized that they could take inspiration from nature. They looked at how the body stores insulin, right? It doesn't just float freely.
No, zinc naturally organizes insulin into these stable, structurally ordered hexamers.
Right. So the researchers wondered if they could create a similarly ordered metal-based nanoparticle to stabilize this tricky S-STING drug and somehow get it past the liver.
And they set up a truly elegant experiment to test this. They took a bacterial S-STING agonist, a very potent drug called CDA. They combined it with a synthetic 11 amino acid chain made entirely of histidine.
Histidine.
Yeah, it's an amino acid that has a very specific chemical ring called an imidazole ring, which loves to share electrons and bind to transition metals. And finally, they introduced manganese ions.
Manganese, so just a standard transition metal on the periodic table.
Exactly, a metal perfectly suited for coordination chemistry. Now, the expectation within traditional nanoparticle manufacturing is that when you mix these things, you just get an amorphous, messy, spherical blob.
Right.
But what happened here basically changed the trajectory of their research.
I found the methodology here fascinating, honestly, because to understand what was happening at a molecular level, the researchers ran Monte Carlo computer simulations.
Yes.
Instead of just guessing, they used these complex algorithms that calculate thousands of random probabilities to figure out exactly how these specific molecules would fold and bond together in space.
Right, it's amazing.
And the simulations, which were then followed by electron microscope imaging, revealed that the ingredients had spontaneously self-assembled into these highly ordered, perfectly uniform, microscopic nanoribbons.
The key word there is spontaneous. It wasn't forced through intense heat or pressure. The chemical properties of the manganese ions acted as a bridge.
Wow.
The metal physically pulled the S-STING drug and the histidine chain together, locking them into a rigid repeating crystalline structure.
That is wild.
And to ensure this ribbon could travel through the bloodstream without getting caught by those liver cells we mentioned earlier, they wrapped these tiny metal ribbons in a very specific ultra-thin lipid coat, and they named the resulting nanoparticle crystal.
Crystal. I want to visualize this because the structural difference from traditional medicine is profound. It's the difference between throwing a handful of loose, messy bricks at a target, which is the free drugs causing collateral damage everywhere,
Right.
versus engineering those exact same molecules to snap together perfectly, like Lego bricks, forming an armored, uniform delivery vehicle.
And the structural order of those snap-together Legos completely changes how the body interacts with the drug. And that uncovers the real mechanism of why crystal succeeds where previous therapies failed. It all comes down to cellular targeting.
Well, earlier we established that the free drug is highly toxic. We need to explore the why behind that toxicity.
So when you inject free S-STING drugs into the bloodstream, they accidentally get absorbed by CD8 plus T cells.
T cells. Wait, but T cells are the immune system's frontline soldiers. They're the exact cells you are trying to mobilize to fight the cancer.
Exactly.
Yeah.
The free drug causes massive friendly fire.
Oh, no.
T cells are highly sensitive to sudden, massive internal spikes in interferon. When the free drug forces the S-STING pathway open inside a T cell, it overstimulates the cell to the point of exhaustion.
Oh, wow.
The T cell undergoes apoptosis, which is programmed cellular suicide.
That's terrible.
Yeah, the drug was destroying the very army it was supposed to command.
So how does the metal ribbon prevent that from happening?
Because crystal is a structurally rigid armored ribbon, T cells simply cannot absorb it. They just don't have the biological machinery to ingest large, rigid structures.
Oh, I see.
Instead, crystal is specifically taken up by myeloid cells, like dendritic cells and macrophages.
The immune system scouts.
Right. They are phagocytes, meaning their entire job is to patrol the body and eat large foreign objects.
Okay, so the scouts swallow the crystal nanoparticle.
Yes. Once it's inside the acidic environment of the scout cell's endosome, the lipid coat dissolves. The manganese bonds slowly break down, and the ribbon unspools, safely releasing the singing drug directly into the scout's cytoplasm.
And the scout cell just handles it.
It easily survives this. It sounds the interferon alarm perfectly, which then recruits a massive, healthy, circulating army of CD8 plus T cells to attack the tumor.
That is brilliant. By just changing the physical shape of the drug into a ribbon, they entirely bypass the friendly fire.
Exactly. And the physiological results reflect that targeted mechanism perfectly. When they moved to animal trials, they tested this in mice with large late-stage melanoma and in a model of spontaneous, highly aggressive breast cancer.
So tumors that have already established their own blood supply and defenses.
Right, very tough to treat. By injecting ultra-low doses of crystal systemically into the bloodstream, they saw complete tumor regression.
Complete,
And not only regression, but durable immune memory.
What does that mean in this context?
Well, the researchers later took those same cured mice and tried to reintroduce the exact same cancer cells.
Oh, wow.
The mice were 100% resistant. Their immune system's T cells remembered the specific signature of the cancer and wiped it out before a new tumor could even form.
That is incredible. And they didn't just stop at mice, right? They also pushed testing into a highly aggressive rabbit model. And these rabbits had massive primary tumors and severe lung metastases, which closely mimics advanced human disease. And systemic treatment with crystals suppressed the primary tumors and drastically cleared out the lung metastases.
Which is huge. But curing mice and rabbits is, well, it's a necessary milestone, but we have to recognize the historical graveyard of cancer drugs that worked in rodents but completely failed in humans.
Right.
Human immune systems, and the immune systems of larger mammals in general, are notoriously sensitive. We are highly prone to hyperinflammation.
And that is the ultimate so what question for this entire field, isn't it? When we look at advanced immunotherapies currently on the market, like CAR T cell therapy, the side effects can be brutal.
Very brutal.
Patients often develop cytokine release syndrome, or CRS. The immune system just spirals out of control, causing massive fevers, dropping blood pressure, and sometimes organ failure. Moving this STING agonist into larger mammals seems fraught with that exact danger.
The University of Michigan researchers understood that risk intimately. Before even considering human applications, they conducted safety and efficacy trials on healthy dogs and non-human primates.
Okay.
Translating a potent immune activator to primates is a massive jump in complexity.
So did the primates experience that hyperinflammation? Did they get CRS?
They did not.
Wow.
The researchers used those same ultra-low doses. We're talking 0.003 milligrams per kilogram of body weight. Crystal potently activated the S-STING pathway in both the dogs and the primates, but crucially, it did so without triggering cytokine release syndrome.
That is a massive relief.
Yeah. The monkeys and dogs maintained normal organ function, stable blood chemistry, and normal body weight. There were just no signs of the runaway inflammation that plagues other therapies.
I do want to dig into the blood chemistry data, though, because the paper notes there was a massive spike in white blood cells.
Right.
Normally, if a doctor sees your white blood cell count skyrocket, they suspect severe infection or toxicity, right?
Context and timeline are everything here. In standard toxicity, that spike remains dangerously high as the body fights a losing battle. With crystal, the researchers tracked the timeline and realized it wasn't toxicity at all.
Okay.
The white blood cell spike was actually the pharmacological proof that the drug was doing exactly what it was designed to do.
How so?
The immune system was waking up, mobilizing myeloid scouts and T-cell soldiers into the bloodstream and then gracefully returning to baseline just a few days later.
Oh, I see.
It was a controlled drill, not a house fire.
That is such a great distinction. Proving you can ring the alarm loudly but safely in a primate's bloodstream is a huge leap. But this deep dive goes one step further.
It does.
The researchers didn't stop in animal models. They actually tested the drug directly on human tissue.
Yeah, they acquired fresh surgical biopsies from human patients who had just undergone tumor removal for head and neck squamous cell carcinoma.
Right.
They kept this human tumor tissue alive in the lab and treated it with the crystal nanoparticles.
But testing on human tissue in a petri dish is inherently tricky, isn't it? Because it lacks a circulating blood supply to bring in new immune cells. Were they actually able to see a real localized immune response within the biopsy itself?
The tissue lit up. Compared to the free drugs, crystal induced a massive immune response, activating the interferon signals beautifully within the existing immune cells trapped in the tumor microenvironment.
That's amazing.
However, the data revealed a crucial nuance. It did not work for every single patient's tumor.
There is always a biological catch, isn't there? What separated the tissues that responded from the ones that stayed silent?
The efficacy of crystal depends entirely on the patient's underlying genetics, specifically their S-STING haplotype.
Let's unpack haplotype for our listeners. We are talking about the specific genetic variation of the S-STING gene that a person inherited.
Right.
Sort of similar to having the genetic variation for blue eyes versus brown eyes, but in this case, it dictates the structural architecture of your cellular alarm system.
That is an excellent way to frame it. Every human has the S-STING pathway, but slight genetic mutations change how the STING protein is physically built. The researchers found that crystal worked brilliantly for the vast majority of human biopsies, those from patients who had the normal, most common variant, which is known as the R232 haplotype.
Okay, so what was the mutation in the exceptions, the ones where it didn't work?
The therapy failed in patients carrying a specific mutation called R232H.
R232H, got it.
In those tissue samples, the alarm just didn't ring. The R232H mutation actually changes the physical shape of the S-STING protein's binding pocket.
Oh, I see where this is going.
Yeah, because the geometry is altered, the CD8 drug released by the nanoparticle just cannot dock properly. If the key doesn't fit the lock, the interferons are never released.
Wow. That structural reality changes the entire clinical future of this drug. It proves that crystal is not a blind, universal magic bullet.
Exactly. And from a clinical standpoint, recognizing that limitation early is a really good thing.
Why is that?
It means that for crystal to succeed in future human trials, it will require strict, personalized genetic screening. Oncologists will need to sequence a patient's immune cells to verify they actually carry the compatible R232 haplotype before administering the therapy.
So it is the definition of precision medicine. We are relying on activating the host's own healthy scout cells rather than relying on the tumor cells.
Right.
Because cancer often purposefully mutates or breaks its own S-STING machinery to hide from the immune system. But as long as the patient's own inherited immune profile has the right lock, this metal-armored Trojan horse can deliver the key.
The sophistication of leveraging the host's innate immune architecture rather than fighting the tumor's constantly mutating defenses is really why this specific approach holds so much promise.
We have covered a tremendous amount of ground today, moving from the basic coordination chemistry of manganese to primate safety profiles, all the way to human genetic haplotypes. If we are going to distill this investigation down for our listeners, I see three major takeaways here.
Okay, the first takeaway is fundamentally a triumph of biomimicry and engineering.
Right.
By taking inspiration from biology and using metal ions to self-assemble drugs, scientists can create structurally stable, uniform nanoribbons that perform vastly better than traditional, chaotic lipid nanoparticle delivery.
Yeah, snapping the molecular Legos together completely changes the drug's entire biological destiny.
Exactly.
And I'd say the second takeaway centers on safety through cellular targeting. Crystal safely achieves systemic S-STING activation because its rigid structure specifically targets the immune system's scout cells, the myeloid cells that love to swallow large objects.
Right, the phagocytes.
Yeah, and by doing so, it completely bypasses the catastrophic friendly fire toxicity that overstimulated and killed off the T-cell soldiers in previous iterations of the therapy.
Spot on. And the third takeaway really looks to the clinical horizon. The translation to human cancer treatment appears incredibly promising, backed by strong safety data in non-human primates avoiding cytokine release syndrome. However, its success will demand precision medicine, requiring doctors to match the drug to the patient's specific genetic immune profile, their S-STING haplotype, to ensure the biochemical key actually fits the lock.
It really brings us right back to the imagery we started with. It is awe-inspiring to consider that the exact same biological quirk, using metal ions to create precise structures, literally the same mechanism that crystallizes the enamel on our teeth, has now been engineered in a laboratory to build microscopic, cancer-fighting ribbons.
It's amazing.
It reminds us how much raw, untapped potential still resides in biology's most basic building blocks.
It absolutely does. And it leaves us with a highly provocative question regarding the future of pharmacology as a whole.
Okay, what is that?
If we can use manganese and simple coordination chemistry to perfectly organize, stabilize, and safely deliver this highly toxic S-STING drug,
Yeah.
what other incredibly powerful but currently abandoned or too dangerous medicines are just sitting on laboratory shelves right now?
Oh, well.
What else could we resurrect and restructure using this exact same metal assembly blueprint?
That is wild. It makes you wonder how many failed clinical trials from the past decade could be completely reversed just by changing the physical geometry of the delivery vehicle?
Exactly.
That is a fascinating thought to chew on. Thank you all for joining us on this deep dive. If you enjoyed unpacking this science with us, be sure to follow and subscribe so you don't miss our next exploration into the unexpected frontiers of research. Until next time.